हो गया क्लास स्टार्ट। कल ने पता है 100 कितना 25 इंसान पता है।
Uh, good evening, all. So sorry for the inconveniences. So, would you guys able to see my screen? Is my screen visible?
Yes,
You
sir.
guys could see my screen, that is Advanced Bioinformatics, your course material.
Yes, sir.
Yeah, fine. Okay, so good evening to all. This is Ajay from Biorack US Center, Department of Biotechnology. So I'm going to handle your class henceforth. Okay, so welcome you all for this Advanced Bioinformatics course organized by EduFabrica. Okay, so I just wanted this session to be interactive session. Okay, hope you guys are from undergraduate and postgraduate, right? You guys are from undergrads and postgrads.
Yes, sir.
Yes. Yeah.
Yes, sir.
Okay. So yeah, someone could initiate the talk, and you guys can tell me how you guys know about bioinformatics and how well you are trained in bioinformatics, or what is your status about bioinformatics. Please let me know. Yes. Anyone? So people are joining. So yeah, tell me. So how well you know about this bioinformatics, a domain called bioinformatics? Or are you guys familiar with it?
Yes, sir.
Someone can unmute and talk, because yeah, this could be an interactive session. We could come across a lot of questions and stuff. So please
A
unmute and
few
talk.
things about molecular docking and all that,
That's
sir.
great. That's great. That's great. So it's a part of proteomics, right? Yeah.
Yes, sir.
So in this course, we're gonna talk about a lot. We're gonna know about a lot. So is my screen changing? You guys can see my screen changing?
Yes,
Yes,
sir.
sir.
Yes. So
Yes,
these are
sir.
the course content. I guess you are aware of the course content. I just wanted to brush it up. Okay. So yeah, I guess you're aware of the course content. So first is introduction to advanced bioinformatics. So bioinformatics is itself, like, a lot of people are thinking that bioinformatics is a dry subject, and there are no much opportunities in bioinformatics. So I'll give a small self intro about me. I'm a self-taught bioinformatician. I learned bioinformatics by myself, and I started doing freelancing services. Okay, so now I'm a part of a government organization, and side by side I'll be doing this freelancing. So this is a best way for a person with a good internet connection and a good laptop could do world-class research. Okay, and can publish papers on. So that is how convenient this particular course is going to be. You guys following me? I guess so. Yes. Yeah. Anyone? Just unmute and tell me. Hello. Yes, someone? Yeah.
Could you repeat the question, sir?
Sorry, it was not like a question. The thing is, I just asked you guys how you guys are how familiar you guys with bioinformatics, and I just wanted to add on something to that point. So that was my question initially. So yeah, you guys are aware of bioinformatics, and now we're going to talk about advanced bioinformatics. So that was my question. Okay. So you guys could hear me? Am I audible?
Yes, sir.
Yes, yes.
Yes,
So
sir.
yeah, I'll just repeat what I told. I'm a self-taught programmer and a bioinformatician who started doing freelancing in bioinformatics, and this is where I realized, and this is where I came to know we could do a high level of research with a good internet connection and a proper laptop what we have at home. So in the stream of biotechnology, this bioinformatics is so convenient for you guys. So in the era of technology, this comes in handy for your research. Okay, so this is what you are gonna learn for the next 1 month. Okay, so yeah, these are the contents like introduction to advanced bioinformatics. In this, we'll be talking about overview of advanced bioinformatic techniques and their application, role of the softwares. There are a lot of open source softwares, there are a lot of tools, and there are a lot of other softwares which can be paid or which can be procured from different sources. And emerging trends and challenges in the field. We'll be discussing briefly about the challenges in the field. So being in the same field for more than a couple of years. So as a young researcher, will be facing a lot of problems, and those problems are like we can overcome those. So we'll to be talking about that. And next is the exciting part, that is the next generation sequencing data analysis. So you guys will be having some kind of gist about. You'll be having some insight on next generation sequencing. Okay. So next generation sequencing and its data analysis will be doing pre-processing and quality control of NGS. So what a data is given by a NGS person is a raw data. So we should know how to interpret the data. We should know how to modify, and we should know how to uh, make it for a person who reads your article, or a person who's reviewing your article, some person, to make it easy for them to understand. So end of the day, it is a computational data, right? So, and we'll be seeing about the pre-processing and quality control of NGS data, alignment and mapping of the sequences. Like we'll be creating alignment reads and mapping of particular data, that is the sequence data. We'll be cross-verifying with all of biological databases. And variant calling, what variant does it comes under? Like we'll under a genus or under a species, we'll be having so much strains and variants. We'll be doing variant calling and structural variant analysis. And in this NGS data analysis, we'll be using some backend based tools. This is something called a backend based tool is called is known to be where you work not on the tools what you see day to day. You will be doing some small scripting in the form of terminal, that is in your command line or in your Unix terminal to run the software. So these are BWA, Bowtie, SAM tools, GATK, all these tools are terminal based tools. Okay. And don't worry, we'll just split it and we'll just have a talk about that. So next one is transport analysis, managing and other stuff. And differential gene expression analysis. We'll be talking about that. Splicing analysis and isoform quantification. This might sound so difficult to you guys, but uh, yeah, someone have raised their hands. Please unmute and talk.
So some students want to join the meeting. They already
Sorry,
have
sorry, I
video.
couldn't hear you properly. Sorry.
So some students want to join the meeting, but they're not able to join.
But I couldn't see them here in the waiting room. If they are there, I could be admitting them. I couldn't see them.
Okay, 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 sir.
Please ask them to retry. Okay.
Okay, sir.
Yeah, please ask them to retry. I'll let them in. Okay. Yeah. So, sorry, we'll be seeing about this splicing analysis and isoform quantification, long read sequence, and data analysis. And there are so much data, so much, sorry, tools associated with these kind of analysis and sequencing. Okay, yeah. And tools we'll be using are DE, Salmon, Flare. Being in an advanced bioinformatics course, there is not much thing needed. That is like not much prerequisite needed, but you guys should aware or you should know about some scripting, basically on the bash scripting, basically on command line, and also some Python scripting. So that will be so easy for you guys to know what is happening here. Okay, so as I told you before, this DC2, Salmon, Flare, all these are backend tools. That is, those tools are operated with a terminal, with the help of the terminal. Okay, and next thing is epigenomics and chromatin analysis. So yeah, people are actively joining. Yes. So epigenomics and chromatin analysis, like we'll be analyzing the DNA methylation and histone modification. This is about the epigenetic modification, what happens when we annotate a DNA sequence, and chromatin accessibility and chromatin confirmation analysis, and integration of epigenomic data into a omics data. Like we have a lot of omics, proteomics, genomics. Uh, yeah, Harsh, tell me. You've raised your hand.
Am I audible, sir?
Yeah, you're audible. Yeah, tell me, tell
So I
me.
just had one question. Just now you said that for understanding this entire course, you should be having some knowledge about scripting.
Yeah, you no need to worry about that. This is not
Means
like I'm just
like
stating a fact.
Python script.
So, sorry, sorry. Yes, yes, yes. Yeah, tell me.
You said I mean about Python scripting and all, but sir, actually I'm from biotechnology background, but I have zero knowledge about this Python scripting and all. So
No
will I
problem
be
at
able to
all.
cope with it?
Yeah, no problem at all. Like, what I was meant to say is, you should know what is Python and you should know what is this bash scripting or terminal. That is the only thing I told. Okay, because we are here to help you out with the scripting and stuff. Okay. What I
Thanks so
meant
much.
is you just should know what are the tools or what are the scripting softwares we're gonna overcome, like we're gonna see across.
Noted, noted, sir. Yes.
Yes, yeah, fine. So you guys no need to worry about that because we biologists or we life science people, we usually look this computer kind of thing in like a halo or like something which is huge, cannot be taught like that. But totally after entering into bioinformatics, I consider it as a myth because it is so easy. And I already told you we are in the era of technology. We have a lot of GPT based models, AI models, and machine learning models. Here, for example, ChatGPT comes so handy for us. Okay, we can use ChatGPT. That too, we have a update on ChatGPT that is 4.0 version. So it is so smarter than the previous version. So we should know how to ask questions. We should know how to extract something out of it. So in that case, we could ask it itself to do a coding, do a scripting. Okay. Apart from that, we'll just teach per the code we come across. Okay, we'll just I'll teach you how to do it. Okay. Yeah, next is structural bioinformatics and protein structure. This is comes into this proteomics where people told about this molecular docking and simulation, right? Like protein structure prediction models. We have a lot of models and algorithms behind it, like homology model, ab inito model, like that, and structure-based drug design. Like we'll be having the protein molecules will be having pockets, binding pockets. So based on that, we can repurpose a drug, already existing drug, or we can just take a drug from any sort of biologically available compound. We can just repurpose it to drug, and that kind of approaches we can learn from here. And software tools like Modeler, Rosetta, and Bioinformatics Studio, Autodoc, Vina. And we'll just have it just about this Schrodinger suits and Swiss suits like that. We'll tell you about those tools also, and proteomic data analysis, like just yeah, mass spectrometry data processing and peptide identification. Peptides are short sequences of proteins, smaller sequences of proteins and small space proteins, and quantitative proteomics methods like label-free and isobaric labelings and and enteric labelings like that, and protein-protein interaction, how proteins interact with other protein, what kind of interaction it has, like pi-pi interaction, pi-pi cation interaction, what kind of interaction it has. And in that particularly will be the leads called Vacuum, Selenizer. These softwares are so interactive. Okay. And next thing is network biology and pathway analysis. Like you will be having, because we are biological organisms, we have lot of n number of networks and pathways running behind the wheels. Okay. So we all know this Krebs cycle, and a lot of metabolic pathways. So we'll be dealing with those kind of pathway analysis, how metabolite is interrelated, or how a protein or how a compound is interrelated with other cyclic manners, behaviors, and pathways like that, like construction analysis of biological networks, protein-protein interaction networks, and what are the genes having in common. All like what is a genome? Like all genes put together forms a genome, right? So in that gene analysis and gene regulatory networks, how a particular gene forms a network with other functional genes like that, and pathway enrichment analysis and functional notation called we'll be doing this with a string, cytoscape, reactome, and itor, and another pathway analysis software called the KEG pathway analyzer. So we'll be coming across all these softwares. Okay. So are you guys following me? Yes.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes. Yeah.
Sir.
Yes,
Good.
sir.
Yes, sir.
Yeah, and next thing will be the 8th, 9th, and 10th will be so challenging for you guys, because you guys are uh, bioinformatics, like biotechnologies or life science people. Yeah, most of them are from biotechnology or from different life science
Yes,
Yes, sir.
background.
Yes,
biotechnology.
sir.
Yes, yeah, that's great.
Yes, sir.
So I'm from a biotechnology background, and I turned into a bioinformatician. So yeah, I guess I'll be telling you what happened and how I turned like that. I'll be telling about those in upcoming classes. And metagenomic analysis. So metagenomic analysis is one of the growing kind of yeah. Shrutika, you have raised your hand. Shrutika, right? Yes. Tell me.
Good evening, sir.
Good evening.
Am I audible to you?
Yeah. Yes, you are audible.
I actually had a question that I
Yes.
am from life science background, and my subjects are chemistry, botany, and zoology. And do you think that I'll be able to cope up with this subject? Or
Very well. You can cope up, because this what you're talking about, this genes, this DNA, central dogma, DNA, RNA, protein. This all we are just training ourselves from school level itself, right? Is it?
yes, sir.
So
Yes, sir.
we're gonna just talk about those stuff repeatedly and eccentrically. Okay, we're going to talk about those stuffs alone, because that is what all put together in a computational form is what advanced bioinformatics. So these terms, these jargons maybe kind of outlandish to you guys, but come on, we'll just be cheaper, and we'll just I'll tell you it is the easiest part. Like there are no particular. Sorry.
Sorry, sir, you are
Yes.
not audible. Hello.
Am I audible now?
Yes, sir. Now you are
Yes.
audible.
So yes. So we are here to teach you, and we are here to help you out with that. Okay. So answering your question, yes, you can very well cope with the thing. It is not depending on bioinformatics, or people are considering, this is a myth actually. People are considering bioinformatics as one of the subjects which is inside biotechnology. So please don't be at that mindset. Okay. It is also a stream of science where
Okay, sir.
you scientists or you science people can cope up with. Okay. Only thing is you're using technology day by day, and we are going to drive you through the other way around, where you can use the technology for this kind of purposes. And yeah, Krishnamurthy, you have raised your hand.
Yeah. Sir,
Yes,
hello.
yeah, tell me, Krishnamurthy.
Sir,
Yeah, yes.
sir, actually, I am doing third year PhD in plant genetic research from NPPGR, sir.
From where?
Hmm. IRA, sir. NPG, National Bureau of Plant Genetic Resources, sir.
Okay, yeah. சொல்லுங்க. Tell me.
சார் எனக்கு வந்து இப்போ நான் வந்து எஸ்எஸ்ஆர் மார்க்கர்ல
நீங்க
தான் சார் ஒர்க்
முடிஞ்ச
பண்ணிட்டு
வரைக்கும்
இருக்கேன்.
try
இப்போ
to talk in English so that others could understand.
Sir, actually I am working in SSR marker diversity analysis only, sir.
Okay.
SSR mark. Sir, I want to learn to like SSR MESA to some other different DEG sequence, which is usually related to
Yeah, XPC softwares, right? XPC 11.
yes, sir.
Yes,
sir.
okay. So this is what you're talking about is all purely into genetics part, genomics and genetics, right? Like cross verification of gene and cross verification of genome. So we do have a specific thing for that. Like it comes under this epigenomics and chromatin analysis. உங்களுக்கு transcriptomic analysis, epigenomic analysis, we'll just talk about that. Okay. So no need to worry about that. Okay. So you're doing PhD, right?
Yes, sir. I'm third year, sir.
Yes. Yeah, very good. And yeah, and anyone else have any question? Yeah, since it's the first day, I'm just giving you just about bioinformatics and stuff. Okay. Yeah, Shane, tell me, what is your question?
Sir, can having a laptop help us in studying this course much
That
better?
is the first thing. Yes, I'll tell you what. You should be having a laptop. Okay. I guess you guys are joined through your phone, right?
Yes.
Joined through your phone. So
Yes.
that might help you now. Because we are just focusing on some theoretical part. This whole course is going to be full theoretical part, and we'll be having some practical part also. But if you are aiming to become a bioinformatician, or if you wanted to incorporate this bioinformatic methods into your projects, future projects or research, something like that, a good configuration is somewhere around i5 configuration, okay, with 500 GB SSD hard drive, or with 8 GB minimum memory, that is RAM. So pretty much all these laptops nowadays are having this configuration, okay? And if you guys don't have this configuration also, I'll teach you some tricks and hints where you can use cloud. You can repurpose cloud, that is cloud computing, to do bioinformatics, okay?
8 GB RAM or 8 GB memory?
8 GB RAM and 500 GB SSD, that is ROM. That is your hard drive memory.
No, for studying this course, a specific course.
For studying this, you don't need much. You just follow the class. Okay, if you wanted to recreate what you learn here, this is one of the best configuration a bioinformatician will have. Okay, like a lot of people will be having i3 generation laptops. That is also fine. Because a lot of tools are open source tools. A lot of tools are web-based tools. So bioinformatics is not that much critical. Okay, the tools which you are talking about is not that much critical. Okay, there are some critical softwares where we'll just show you in a well configured laptop. We'll just show you other stuffs. I'll teach you how you guys could do it, like you can be having a hands-on.
So for
Okay,
practicing these skills, we should have at least a basic laptop.
A basic laptop is much needed, because anyway when you're doing with your own hand, it'll be helpful. Yes.
Okay, sir. Okay.
Yes.
Thank you.
Yeah, thank you. And next question, someone have raised a hand. Rithi, right? Yeah, Rithi.
Good evening, sir. Am I audible?
Yeah, yes, you are audible.
Sir, once when I went to the syllabus, I saw about the visualization and data interpretation thing. Like there has been several software tools that we are using for that. So you will be giving the practical knowledge about it as well, or just the theoretical one?
Yeah, since this is just an 1 hour class, we may brainstorm a lot. We may brainstorm and we can discuss a lot. But I'll try to just squeeze in some kind of hands-on or some kind of training that would help you out. Or else I'll just show you from my side so that you will be aware of it. Okay. If the software is so light, or if software is pre-built and available in the web, I'll just share the link and I'll ask you to do. Okay, so you guys don't worry about that.
Okay, sir. Thank you.
Thank you. And Harshik, you raised your hand.
Yes, sir.
Yeah,
I'm audible,
tell me. Yes, you are.
Yes. The tools we are going to use are open source, or These are paid
Some
tools.
some tools are open source tools. Some tools are paid tools, which comes under
Okay.
some stipulated time of free usage. You can use the tools for 1 month or so with your official mail ID like that. Okay, and
Okay.
we do have alternate for those tools as well. Okay, so no need to worry about that.
So most of
And
the tools have alternative open source tools, right?
yes, yes, yes. We can just come the other way around. Like if you're just gonna use some visualization tool, which is a paid software, with the same configuration or with the same algorithm, there'll be another software. Okay. So, and I told you about Python, right? Python has a lot of libraries pre-built within that. So Python and R programming, R Studio, right? All these scripting languages have their own inbuilt libraries where the bioinformatician mostly rely on. We create tools with that. If we are not having some sort of tools which is not working properly, we'll create our tools. Okay. uh, in that case, will like why bioinformaticians are running behind Python is Python is not like other computer languages, computer language models. It is so easy to learn, and it is like literally talking to a computer. Okay. For example, I'll just say you might heard of this C++ and Java. So for printing a statement in C++ Java, it may take around 4 to 5 lines of code just for printing a statement called hello world or your name. But in Python, you can just do it in one line. And as I told you before, we have a lot of AI models. We have a lot of machine learning models and ChatGPT like that. We can take the help of those models. Okay, this is where BioPython has born. BioPython is where we biotechnicians can use Python for annotation, for visualization, and for processing the data. We'll talk about that. We'll talk about that. Okay. Yeah, Soumya, you raised your hand.
Yeah, sir. Good evening.
Good evening. Good evening. Yeah. Yes.
So actually, this is like 45 days course, right? It's with internship and training. So
Uh, Okay. Yes, this is a 1 month course. This is a 45-day course. Okay.
yeah.
And I'm just a person who teaches this, and other stuff's are happening back at the background. So if you want any clarifications regarding the course and stuff, I'll let you know later. Is
Okay,
that fine?
okay, sir.
Because
Thank
this
yeah.
is my first class. Since this is my first class, I have to like I'm training with you. I'm getting trained with
Okay,
you guys. Okay.
okay,
So
okay.
yes. Yeah. And yeah, Varshini, you have raised your hand.
Good evening, sir.
Yeah, good evening.
Sir, you said a lot about Python scripting and Python language. Is it necessary to know Python before we take up this course?
No, 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 no. It's not like necessary. I just gave you a hint about the thing. It is not like you should know Python and R programming, but it is good to know because a lot of this software is like here, the module where I'll show you. Wait, most of the tools here are built on Python. Okay.
Okay,
And
sir.
that's why we should know some just about Python. I'll just show you what are the tools. This bioconductor right here on 10th integrated bioinformatics and multi-omics data integration. So the tools here is bioconductor and omics integrator. So these are packages of Python. So these are packages of Python, and this BioPython and other tools like Autodoc, Vina, Autodoc, all these are made with Python. That is what I mentioned. So it is not like you should know Python to take this course. I'm telling I'll give you information regarding Python so that it'll be helping you with this course.
Okay, sir. Got it, sir. Thank you, sir.
Thank you. And yeah, Firoz, you raised your hand. Raise your hand. So yeah, I guess we
Hello.
are having some brainstorming session here. Yeah, yes, tell me, Firoz.
Uh, Sir, my question is, sir, after completion of this course, we are able to publish paper in this domain, bioinformatics.
Yes, absolutely. So recently I've published a paper on World Journal of Pharmaceutical Research. I just did only docking and screening, okay, with my laptop, and I published a paper. and it has an impact factor around 8.6 something. So that is what I told earlier. Bioinformatics is a domain where you can have a laptop and a good internet connection. We can do a world-class research, or you can do a well kind of research in this. There's something called an in silico research itself. In silico research is what bioinformatics research is. So that is why we have a lot of demand for this bioinformaticians.
Okay,
Yeah.
sir. Sir, can you share with us your means, sir, your publication?
My article? Yeah, I'll definitely
Yeah,
do
yes,
that.
sir. Yes, sir.
Okay.
Yes, sir.
I'll try to do
We'll
that.
get
Okay.
some idea from that.
Sure. We'll definitely do that. And I'll show you how you guys can also do that. Okay. So,
Okay.
and due to the time constraint, we should just complete this course on time, but we'll be helping you out. Okay. From our side, we'll be helping you out. No need to worry. So yeah, I'll just complete this alone. Machine learning and deep learning in bioinformatics. This is going to be an exciting part when you are eager to learn it. If you guys are so eager to learn about machine learning and deep learning algorithms, this is going to be so handy for you guys, and you can just do lot of models. You guys can create models, deep learning models and deep learning algorithms, and you can get intellectual property for that, and you can be publishing it, and people can use that as open source softwares, and a lot of things are there. So machine learning is one of the aspect of bioinformatics. So it's an exciting part, like application machine learning and deep learning in bioinformatics, because we bioinformaticians come across a lot of data, like GBs and GBs and terabytes, sometimes terabytes and terabytes of data, right? And to analyze and to automate stuff, uh, we can't do it all alone manually. So we should be relying on this machine learning models and deep learning models. So prediction of protein structure and function can be done using machine learning. And disease classification, biomarker discovery can be done using deep learning. When you have a lot of data in the database, you can cross verify that disease database, and you can classify data, and you can just identify the biomarkers, specify biomarkers for the disease. And here basically we'll be using scikit-learn and TensorFlow and PyTorch. It's also a Python module. Yeah, someone's joining. Yes. So, and next thing is integrated bioinformatics and multiomics data integration. Multiomics data integration is where this genomics, proteomics, transcriptomics, metabolomics all put together, and we come up with data. So methods of integrating multiomics data, genomics, transcriptomics, proteomics, metabolomics, like that, plantomics, and there are a lot of omics, and introduction to molecular signatures and disease subtypes. So here, as an example, we can say diabetes as a disease, and its subtypes are like type 1, type 2, and we have type 1A, like that and all. And softwares we'll be using in integrated bioinformatics are bioconductor. It is also one of the packages in Python, and omics generator. And on next, visualization data interpretation is also a good thing, where you visualize the data, like all the biological data. It will be in the form of raw data, and you should process the data and visualizing it, and making it presentable for your article or research is what we do here. Visualization data interpretation. Like here, data visualization techniques of biological data will do interactive visualization. Interactive visualization where you find the bonds between protein and its ligand, or protein and the thing, and you'll be doing simulation, and you'll be creating movie files of the simulation, molecular simulation. That is where you simulate a molecule, a protein or a ligand or enzyme in in a molecular level. In a molecular level, you'll be applying its parameters. Like you'll be having software. You'll be having softwares for that, like Chromax, Schrodinger, Mastro is there, and Schrodinger's a lot of software, like lamps, a lot of softwares like that. And you'll be creating movie files. And the software tools here is ggplot. ggplot2 is one of the packages of R programming. Plotty is also one of the R programming software. We'll be plotting the data, and IGV is interactive genomic viewer. So we'll be talking about these stuff. Okay. So you guys have question? Varshini, you have raised your hand.
Sir, you said you moved from biotechnology to bioinformatics. What
Yes.
what resources did you refer to, like in the beginning when you like started
Uh,
learning
like
bioinformatics?
a couple of years ago, I just had YouTube and a good internet connection. That is what I had. Okay. So, and the urge to learn stuff. This is I just don't want to like demotivate or degrade you. This is what I had, and uh, like you have an idea. You biotechnologists will be having an idea, right? So the only thing I had is keep thinking out of the box capability. Okay, what if why scenarios you should be asking to the same principles or approaches what you're learning in biotechnology. Okay, so that is how I turned. Initially, I do practice cybersecurity and ethical hacking. That is where I ended up to know about Python and other programming languages. So I too know this bioinformatics, like Python. Python is used in bioinformatics. And initially, I attended several workshops around, and I attended several hands-on trainings around, and I learned myself. So only thing is, there are good data visualization books and bioinformatics books to refer, and there are a lot of video tutorials. And NCBI has the greatest resource. NCBI's PubMed has the greatest resource, and Expercy is a suite of bioinformatic tools where you can learn about a lot of bioinformatic tools. Swiss tools. It's called Swiss tools. Like Swiss Army knife. Swiss Army knife comes handy when you're in a survival situation, right? So Expercy is the Swiss Army knife of the bioinformaticians, where you know a lot about bioinformatics. So, and one more thing, I practice a lot. I have a good laptop. Like it is not like a very expensive or very well configured laptop. It's just a normal laptop to do day-to-day documentation activity. But you can just download some tools and you can just practice, because all these have data pre-built. Okay, you have a lot of databases. Like NCBI is a database where you can just go and play around with nucleotides, genes, genomes, and Bankit will be having a lot of gene annotation data, SRA, RNA data like that. And PDB comes handy for all the proteins and enzymes, cofactors like that. So everything is available in the web. Okay, and to make it easy for you guys, we are just providing you this course. Okay. So only thing is, if you have that urge to learn, then it's a jackpot for you guys. You can very well become a good bioinformatician. Yes, I guess I answered your question, or
Yes, sir.
yes,
Yes,
Vashini?
sir. Thank you, sir.
Yes. Thank you. So that is what we have discussed the course content now. So from tomorrow onwards, we'll just start with our first module, and what we'll know what is. Do you guys have a basic bioinformatics knowledge? Just answer no. Just one of you.
Yes.
Yes, sir.
Yes, sir.
Yes, sir.
So
Yes.
being a biotechnologist, you should be having, like many of you guys will be having a subject, right? Subject or as a
Yes.
paper, you'll be having bioinformatics.
Yes, sir.
No, sir.
Bioinformatics
Yeah,
was one of the subject in biotechnology.
yeah, it is. It is. And yeah, Saurabh have raised this question, his hands. Saurabh, right? Yeah,
Yes, sir.
Saurabh.
I'm from BTech. Yes, I'm from BTech Bioinformatics. Sir, what
That's
are the
great.
areas where we can do freelance?
Yeah, freelancing. So let's talk about freelancing. I did freelancing on proteomics, where there are so less bioinformatician does this, where doing stuffs on molecular docking and molecular simulation. So molecular docking and molecular simulation is the best freelancing ability or freelancing thing you can do. And next thing is NGS data analysis. That is what you're going to know next. So where this particular freelancing comes in place, where you're doing a project, you will be doing some generation sequencing, or you'll be doing some auto docking or something like that. You'll approach a person, or you do it by yourself, or you'll download the data from the databases, but you don't know how to interpret the data. This is where main picture of the bioinformatics comes in. Okay, so nowadays in research, this bioinformatics is becoming a crucial role. Like you can't just publish a good paper, high impact panel impact factor journal papers without with less chapters, with only in vitro or in vivo studies. So in silico is taking a crucial part. So according to freelancing, You can do freelancing on proteomics, like protein-protein interaction, protein-ligand interaction, like that. And cake pathway analysis. This is pathway, network biology and pathway analysis. You can do freelancing on that. And you can do very good freelancing on next generation sequencing data analysis, where people will be getting the data from a sequencing company, but they don't know how to interpret the data. They'll just get the data and they'll include that data into the paper, incorporate the exact data without the change in a full stop or a comma. They'll just incorporate the data into the paper. So this can be comes in this is so handy that you can be processing the data, and you'll be breaking it down into small bits and pieces, and you can just make it into a visualization form, or can make it easy for the person who reads the paper or who understands the research very well. So these are the area, like in this particular module you can do freelancing on all these things, because it has its own tools, and people out there, they don't know this much tools are existed. So if you guys learn these tools, and if you know a proper target audience, or if you know proper market on stuff, you can just focus on that. You can identify the market, and you can it is not like identifying a market. You can just help your friends to, or you help your faculties to do it. Okay, that is how you develop a network. And that is how I developed a network. I used to help my faculties, I used to help my friends with this, and that is how contact developed. Okay. Yes. Anything else? Danush. Yeah, you've raised your hand, Danush.
Yeah. Good evening, sir.
Yeah, good evening. Tell me.
Sir, I'm from Bachelor of Pharmacy. I have did my Bachelor of Pharmacy. So I plan
That's
to move to
great.
Bioinformatics.
That's great. That's great.
So what are the, preliminary things should I go through?
So you know very well about drug, right? Drug.
Yeah,
So
sir.
since from
Yeah.
a pharmaceutical or pharmacology background,
Yeah, sir.
you have a good scope on bioinformatics under structure-based drug design or computational drug designing. Okay, where
Yeah, sir.
you
I have done
repurpose.
my project. Yes, sir.
Okay, that's great.
Yes, sir. I have
Yeah.
done my project
So
in
you know
molecular
you
logging.
have the idea about it. Yeah. And so what's your question
Yes,
then?
sir. Yeah, sir. But apart from that, biotech idea should be, should I incorporate with it? Because I was lagging biotech ideas, so concepts.
So there is nothing
So
called this is the thing. Like you guys are seeing it as subjects. Okay. You should see it as a knowledge, not as a subject. If you guys see it as a subject, you'll be taking it as a subject. Okay. Bioinformatics is not a subject, biotechnology is not a subject. Okay, this all put together is life science. We are life science people. So biotechnology approach and aspects is not outlandish for you guys, because you're dealing with organisms. Okay, you're dealing with drugs. You're dealing with uh, uh, like kind of a biological organism, right?
yes, sir.
So it is not outlandish. So biotechnology is a knowledge, or it is a collection of knowledge, or collection of methods you can learn from biotechnology. It is not like you don't see it as a subject. Okay. So
Okay.
and here in this course will help you out with that. Here this course will help you with the jargons, or some specific type of like words that which you didn't know, or specific type of techniques which you didn't know. Will help you with that. Okay.
Okay.
We'll provide you the
Thank you, sir.
resources. Yeah, thank you.
Okay.
And yeah, Shailaj, you raised your hand, and Kaidim,
So,
you raised your hand.
good evening, sir.
Yeah, good evening. Tell me.
I'm Sailaja. So currently I'm pursuing my undergraduate program in genetic engineering from SRM University, sir.
That's great.
So
Yeah.
I just completed my second year, so I'm just gonna step into third year. So the thing
Mm-mm-mm.
is, my college doesn't provide the bioinformatics courses. So like I literally have zero knowledge about bioinformatics. So will I be able
Okay.
to follow your lectures and also?
Yeah, you would, because you're from a biotechnology background, and you're gonna know that a lot of things which we discuss here is much more familiar to you. Okay, there are a lot of
Okay.
familiar things we're gonna discuss. Only thing is, we're gonna discuss that in a computational perspective. Okay.
Okay, sir. Okay.
So
Okay.
these
Okay.
tools and this is just an aid for your learning. Okay. These tools and this
Okay. Okay.
programming are just aid for your tool aid for your learning. Okay.
Okay, sir.
So
Okay.
if you're having zero knowledge also, it's not a problem. Anyway, we are gonna learn together. Okay.
Okay,
Yeah.
sir. Okay, sir.
And someone
Thank
else,
you.
yeah, they've raised your hand.
Good evening, sir.
Yes, tell me. Who's this?
Good evening, sir. My name is Kaidem Kennedy Singh, sir.
Yeah, yes. Tell me, Kaidem.
My question is on as we go through the course and we make for this paper publication. So without the wet lab studies also, can we go for the good publication, sir?
Yes, that is what I talked about. Yeah. Without any wet lab experience also, there are good in silico papers around, which is published
Yes,
in
sir.
Springer elsewhere and good Scopus journals. Okay.
Oh,
It is
okay,
just the
sir.
quality of the paper and which is just the method and methodology is what we approach. Okay. There's something called an
Okay.
in silico paper, right? So
Yes, sir. Yes, sir.
it is there. It is there. It do exist. It does exist. Okay.
Okay.
So that
Yes.
is what, like, with a good internet connection and with a good laptop, we can just publish a papers. We can publish a lot of
Okay.
papers, especially on this proteomics, genomics, circos, like that. Yeah, you can do.
Okay.
So I
Okay, sir.
guess the meeting is gonna end now because 78 was the time. Until that, we can discuss. It's not a problem. Okay. Yeah.
Thank you, sir.
Partika Ghosh, you have raised your hand.
Hello, sir. Am I audible?
Yes, you are audible. Tell me.
Actually, I'm just a first year zoology student, but I'm
Okay.
really interested in bioinformatics. Like, I've been
Okay.
researching a lot about it. So that's why I
Yes.
wanted to do this. So,
Okay.
do I need to do some beforehand research or study before I go into this course?
So, were you there yesterday? Were you there yesterday? Were you
Yeah, I
present in the
was,
class?
but it was a
So,
bit.
okay, since it was the first day, I couldn't understand a lot, right?
Yeah.
So this is an advanced course, Introduction to Advanced Bioinformatics, but there's the thing called so advanced. So the word advanced means here you're going to learn extra. That is what it means. So it is not there's nothing other meaning other than that. Okay. So being in a zoology, first year zoology, no prior knowledge is needed because we're gonna help you out. We're gonna teach you from scratch from A to Z. Okay. Only thing is you should know what is bioinformatics. Bioinformatics is a stream of science or stream of biotechnology where you use computational tools, softwares, scripting, and databases to make your research easy. Okay.
Okay. Thank you,
So
sir.
that is what bioinformatics is. It's not like computational. Like computational part in bioinformatics is so easy. No, don't think about those computer people who are doing this BSc computer
No,
science, MSc
actually,
computer
I
science.
am doing a coding course on Python, and I already
That's
know
great.
Java and HTML.
Oh, so it'll be easy for you then. So
Okay.
you know just about Python, right?
Yeah, I
So
know a little bit because I'm doing the course, but Java and
you know how
HTML are
easy
my.
this Python is. So
Yeah, it is very easy comparatively.
yeah, so yeah, I will tell others to will teach those stuffs to other people too. But I will teach Python to other people too. So it is easy. Python is easy. So once you know the flavor of Python and this bioinformatics is gonna be so easy. It will be a cakewalk for you guys. Okay. So
Okay,
first of
thank
all,
you,
I'll
sir.
just tell you, don't be afraid. Don't think that you couldn't cope up with this course, or don't think that this is not your cup of tea. This is for you guys. It is so mandatory. I tell you, this is mandatory. Okay. Because you're going to become the future researchers. Okay. Most of you guys will pursue your career under research, right? So you should know the flavor of bioinformatics as well. Yeah. So this helped me a lot in my career. So I'm gonna share some of my things to you guys also. And this is how I ended up being a good researcher. Okay. So people are just joining. I don't know why they are joining now. Okay. Anything else? Anything else? Anything else? Anyone?
Sir, good evening, sir. My name is Navya Gupta.
Yeah, Navya. Tell me.
Sir, tomorrow
Sorry.
also the timing will remain same or it will change each day?
So I guess I don't know because we people who are teaching multiple courses, we are just using the same login credential. But we'll make it happen. Yeah. What is your convenient time? Like for all of you, I'm just asking all of you, what is your convenient time? Because some people have demanded earlier, some people have demanded later, and some people have demanded in the morning. But I don't know how well it works. But we'll see to that. We'll set a time frame where all the people enrolled in this course can attend. Okay.
Okay, sir. Okay.
So just do one thing. Tell me the common timing. Tell me some common timing that 7 to 8. 7 to 8. A lot of people are telling 7 to 8. It is fine.
Yes, sir. 7 to 8 is fine, sir.
Yeah, because you guys are just trailing up a day. Okay, you will be doing your stuffs in the morning till evening, and you guys will be tired. And I know because you guys will be tired, and you don't have minds to take this input. This is an eccentric input that your mind would process. So in the later day, it won't process so much. Okay, just a psychological thing. 7 to 8 is fine. A lot of people are telling 7 to 8, and some people are telling 8 to 9. Okay, we'll see to it. We'll see to it. We'll just make things happen. I'll just stop sharing my screen so that I could focus on your chat. Yeah. So yes, the end of the session, or else half of the time will be wasted in the question. Yeah, Harsh. We'll do that from tomorrow onwards. We'll save the time for query sections. Since this is the first day, I just wanted to interact with you guys. But tomorrow onwards, when we start the session, we'll have a stipulate time frame for at the end of the session. We'll answer you. We'll answer your questions. You can just note down your questions. We'll answer it later. Okay, is that fine?
Sure, sir.
Yes, yes. So
Sir.
we can stop me anytime and can ask questions. Not like ask questions. Like you can if you have any difficulties, you can start me at the bit. But apart from that, if you have a valid question that you can it can be answered later, you can write it down, and or else you can put it in the chat box. You can and I can answer that, or we'll just discuss. Yeah, since we have just 1 hour time frame or 45 minutes time frame, it is tough, but we'll stick to the time frame. Okay, it's not a big issue. Okay, so 7 to 8 is fine. 7 to 8 is fine. A lot of people are telling 7 to 8 is fine. I think I'll just go with this, or I'll discuss with my I'll discuss with the people around here, and we'll stick with some timing which is so convenient to all. Okay, is that fine? Yeah. Okay. So I
Yes,
could see a
sir.
lot of people telling 8 to 9 and 7 to 8. So yeah, it falls in the same time limit. Okay, fine. So you guys have any other questions or any other thing to say? If you guys have any other questions, please let me know. Please let me know. So would you guys able to follow me? Like what I told you guys able to follow this, or should I change my approach? Yeah, you'll be getting the recorded lectures. The videos are recorded, so you'll be getting it. Don't worry with that thing. Since you are there already in this chat, you'll be able to access the thing, or else we'll share the video or the recorded stuffs to you. So I should be a little bit slow. Not able to follow me. Okay, I'll be slow. Okay. Only thing is, yeah, I've been instructed to complete this within 1 hour. That is the only thing. Okay, so just kindly bear with me, but I'll be a little bit slow. Okay. So tomorrow we'll start with the first first module, that is Introduction to Advanced Bioinformatics. We'll just have an overview of it, okay? Because I could guess that a lot of people are zero to minimal knowledge in bioinformatics. So we should start from scratch. So we'll start from that. We'll start from the introduction. Then next we'll jump on to the other consecutive or other respective chapters. Okay, I'm a final year student and have. Yeah, we'll be sharing you the PPT if you're doing it. And Harsharan, yes. I couldn't get you. Like you have you know what is bioinformatics and you've done projects on that? Harsharan?
Yes, sir.
Is that what? Okay, okay. So you know what is bioinformatics well, right?
Yes, sir. I had already done, and I am a final year student in engineering in biotechnology.
So you worked on protein engineering,
Yes, sir.
proteomics.
My last semester project was on protein engineering.
Okay, okay,
Yes, sir. From
okay.
the last 2 semesters, I'm working in bioinformatics,
That's
and I'm
great. That's great.
really interested in this.
So we do have bioinformaticians here. That's good
Yes,
to
sir.
go. Yeah. When I was pre-final year student, had the same question, but I must bioinformatics is a really good approach in future. Yeah, it is. It is a good approach because everything are becoming computational these days. Okay, people are running behind this computer softwares, computers, and innovation in AI models, machine learning models. So we should also run with them. Being a licensed students or being a licensed people, we should also show our people can able to do that stuff too. We could give tough to them. Yeah, PPT can be shared on WhatsApp group or else in the forum here. We'll share the PPT in the forum here, and the syllabus also will share. Okay. Do so. And someone have asked for the book, right? So there are list of books I could suggest you. Okay, I'll just show you. Wait. So there are, since bioinformatics deals with a lot of computational approaches, there are good books for scripting called Bioinformatics with Perl, Python, Java, like that. You have good books on that. You want a good textbooks? I could prefer Bioinformatics by David Mount. Bioinformatics by David Mount. Just type in the chat.
So bioinformatics by David.
David Mount, Mount. A very detailed book it is. Okay. Like bioinformatics and bioinformatics analysis, sequence analysis. You can go for this book. Okay. And another book called Introduction to Bioinformatics Algorithm. Introduction to Bioinformatics Algorithm. Just a minute. So it is, I don't know who the author is, just with the author. Yeah, I found it. So an introduction to bioinformatics algorithms by Neil C. Jones. This is also a good book to go. Okay. And another book by Robert Gentleman. Okay, I'll just. So this particular book talks lot about R programming, bioconductor, and other computational biology stuff. Okay, Robert Gentleman. So you can follow these books, and I'll be sharing you other details or other books which I find it is easy for you guys to go through or refer along, and other web sources that is easy for you guys to cope up with. I'll just attach it here later in the other upcoming classes. Okay. This is my research article. Just a minute. So let me check whether it is there in Scholar. There. So you, all of you are attending through your mobile phones? Mobile phones? Are you guys using mobile phones still?
No, sir. I'm
Yeah.
using laptop.
Sir,
Some
I'm
of you
also
are.
using laptop.
Okay, that's great then. That's great. So this particular thing is published on World Journal of Pharmaceutical Research, and it just has impact factor of 8.4. Okay, this is a recently published paper on cardiovascular disease. I just did molecular docking and admit profiling. Okay, I'll just put you the link here. So this is the link to the particular journal, the recent publication of mine. But I worked on other articles, other literatures at the back end because I was doing freelancing. I used to work on a lot of articles. I just provide them with the analysis and I'll give them the data. Okay. But this is what I published recently, like worked on recently. So I just wanted to show you this. Okay. Yes. So anything else that we missed discussing today? I guess nothing. Is the link accessible? Could you guys open the PDF? Is it accessible?
Yes, sir.
So this is one of the approach what I underwent, and this is the same approach where we provided some internships to our college students. So you can go through the article. You can just see I just done only molecular docking and admit profiling. And since we have other bioinformatics people here, they also some of the people they told they know molecular docking. So I could see that they could easily make it out. Okay, so this is not only molecular docking and simulation or admit profiling is not only the bioinformatics part. There are other bioinformatics part which is easy, but this is the most easy and reliant one, like reliable one. Okay, so yes. Anything else, guys? So I just don't want to grab or steal your time, your evening. No, this bioconductor is not related to biosensors. So bioconductor is one of the packages in Python. Okay, bioconductor is one of the packages. We have a lot of packages, and one of the package in Python is called BioPython. One of the package in R programming is bioconductor. Okay, it is not related to biosensors. Okay, because we are dealing with bioinformatics, right? We are not dealing with any sort of hardwares here. Okay. I'll just put that link to bioconductor link in the chat box. If you guys want to have a look, you can just see that bioconductor is not related to some kind of hardware or thing, biosensors, but it is a package. It is a collection of packages that we can use for mm, use by sorry, yeah, used in R programming to visualize the data. Okay, is the MD simulation is done by you or? Sorry, I couldn't get this question. Harsharan?
Sir, I just want to ask the molecular docking simulation is done by you or
I did.
any third
I
party is
personally did
involved?
in my laptop, the same laptop what I'm using now. I used Autodoc. So it was just a blind docking approach. Blind docking approach is where you cover the whole molecule with the grid and you run the ligand in multiple places, right? So I did that. I did that simulation. Okay. But since they are just asked only for the lower visualization, we just provided the visualization, the pictures from Autodoc, but we are thought of going to give them a publication quality images from Discovery Studio and Biomol, but they just told the other people, other colleagues with the paper, no, they told this is enough, so we just stopped it there. And this particular paper was just constructed within a week. So just adding a point there. This particular paper is we just completed within a week. Okay.
Okay, sir.
And I didn't take much time, like months and months of time. We sat all together for 1 month and we completed the thing. Okay. So anything else? Shall we wind up the class? Okay,
Yes,
I'll be
sir.
a little bit slow. I'll be
Yes, sir.
I'll be trying to
Sir,
yes, Krishnamurti.
Sir, can you share that link in WhatsApp, sir? Because I joined through phone, I can't download it.
You can't access the chat, meeting chat.
Yes, sir. No, sir. That paper I can't download, sir. Can you share that
The
link?
paper. Okay,
Wow.
okay, okay. I'll
Yes,
share
sir.
that. I'll share the thing. Okay. Yes, yes.
Okay, sir.
Yeah. So you guys can tell me if you are facing any difficulties. So I'll be your instructor henceforth, and yeah. If you guys want to tell anything else, you can just tell, or we can end the meeting. Okay, is that fine? Yes, please answer.
Yes, sir.
Okay. So thank you so much. Thank you all for joining here. Okay. So it's good to you. Like it's good. It's so good that I got an opportunity to interact with you people, and I'm glad that I'll be traveling with you for another 1 month or so. So it's happy learning. Okay, let's learn together. Okay. Please come up with questions. Please come up with a lot of questions because you people are active researchers. Okay, and it was a good day. Okay, thank you all. Thank you all for joining.
Thank you, sir.
Yeah, thank you. Thank you. Thank you. Thank you so much. Let's end the meeting. Yeah, we'll
Have a
end the
good day, sir.
meeting now.
Yes, sir. Yes.
Yeah, have a good day, guys. Have a good night. Yeah, thank you so much. You all can leave now.
Thank you, sir.
Thank you. Thank you.
Thank you,
Thank you
sir.
all. Thank you. So I'll close the meeting. I'll close the meeting for everyone. I'll end the meeting for everyone. Yeah. Thank you, guys. Thank you.